All right, this next chunk, we're talking about muscle excitations. We talked briefly about this, kind of the gist of it. Now we're going to get into exactly how it happens, okay? So this is kind of what we're going to look at, each of these different components, each of these different steps. These are a little bit kind of zoomed in, so I have a couple of other images that we're going to look at that are a little bit more kind of zoomed out, a little bit more big picture of these. So when we talk about excitation, we got the nerve signal, the action potential coming down the nerve signal, and it gets to this whole region. And this whole region is what we're going to call the neuromuscular junction, okay? Because the neuromuscular and the junction is where they meet. So the very first thing that happens is that as the action potential is traveling down this axon and gets to this axon terminal, you have voltage-gated calcium channels, okay? And these calcium channels are located on the walls of the axon terminal. So kind of all through here on the sides like this, you have these voltage-gated calcium channels. As the signal comes down, remember where that action potential is changing, that we're going from, as we're depolarizing, we're going more and more positive, and as we get more positive, these calcium channels open and calcium rushes into the neuron cell. Inside of the cells, these little green bubbles that are all down here, we have acetylcholine. The acetylcholine is being held inside of these little balls called vesicles. Vesicles are kind of like these, you think of them like bubbles, and they have like a mini membrane. They have a membrane wrapping around them to keep in the chemical that they're holding. In this case, it is acetylcholine. So the calcium that gets brought into the cell from those voltage-gated calcium channels are going to interact with the vesicles, okay? And it's going to cause a reaction to occur where it causes the vesicles to actually migrate down to the very, very bottom of this axon terminal, and it's going to help signal them to start to cross over and to head towards that neuromuscular junction, and in this area it's called the synaptic cleft. Another thing you might see here that ACH is referred to as a ligand. Remember, a ligand is going to be any sort of chemical or something that is going to cross and it's going to bind to a receptor. So that's going to be sort of the next step. Before it's able to cross and bind, though, it has to get out. It has to go from the nerve cell into this empty space. The way that it does that is through a process called exocytosis. There's a short little video here I'm just going to play that goes over exocytosis. So we have this vesicle, so imagine that this acetylcholine is inside of this. Sometimes the vesicle will be called a vacuole. Vacuole are just larger vesicles. So imagine in the cell we get the signal from the calcium channels, from the calcium rushing in, and that is going to cause it to migrate to the end, and that the membrane of the vesicle is going to fuse with the membrane of the cell. So in this case, it fuse with the membrane of the neuron. And as it fuses, it literally is going to fuse and then open up, and allowing everything that was inside to come out of the cell. And that's what exocytosis is. Exocytosis, it's to exit the cell. And those synaptic vesicles, the holding the ACH, acetylcholine, is going to be released into this area called the synaptic cleft. And let me actually pull kind of a little bit of a bigger picture. So here we have this is what the neuromuscular junction looks like, kind of a little bit bigger. Okay, those are our voltage-gated calcium channels right there. In our axon terminal, this is all these little red dots are acetylcholine coming into this area, which is known as the synaptic cleft, this area in between the neuron and the muscle, the muscle itself, is the synaptic cleft. This edge of the muscle that's right here is going to be called the motor end plate. And on the motor end plate, you have these ion channels. These are going to be ligand-gated cation channels. Okay? So we see, so the ACH gets across, and it comes and it binds onto these receptors. The acetylcholine binds on these receptors, causing these specific channels to open. And that was right here, it's talking about the end plate. It's these ligand receptors are on the motor end plate, and it diffuses across the cleft, which is the space in between that neuron cell and the muscle cell, between the circle and the sarcolemma. Remember, sarcolemma is the membrane of the muscle. So these ligand-gated cation channels are going to open. So what makes these different is a lot of the channels that we've talked about up to this point only let one thing through, either one thing in or one thing out. It's kind of what we've seen so far. These channels are special. These channels are actually going to allow sodium to rush in and at the same time let potassium flow out. Now, overall, you're getting a lot more sodium rushing in than potassium going out, but still, it's called a cation channel because it's allowing cations, both cations, to flow. Remember, a cation is just a positively charged ion. And for the purposes of this class, we only really get into the sodium and the potassium, but technically, yes, also calcium and other cations do flow through some of these channels as well. They're not, it's like out of the 99%, like 1% might be calcium that's flowing through here. So we just focus on the sodium and the potassium entering the cell and the potassium leaving the cell. And what this does is it changes the voltage at the end plate. Remember, the end plate is this part right here. So here at this motor end plate, the voltage, that potential, we call it, the voltage or the potential, goes starting at -90. In the previous graph, in the previous lecture, we saw it at -80. It's in that range, -80 to -90. You'll kind of see numbers fluctuate. Me personally, I would say neuron, set it at -70. Skeletal muscle, go with -90. Okay, millivolts. Those two numbers, I said, those are the best ones to kind of go with. Okay. So as we get more and more positive, what actually happens is you see how this motor end plate is here, but all of this across is all connected. The sarcoplasma inside, just like the cytoplasm, it's all connected. And so as we get cations flowing into here, these cations are going to drift out and out and out and out and cause that change in the potential, that voltage, to continue to extend all the way here to the edges where we have voltage-gated sodium channels. We have voltage-regulated ion gates. We have these voltage-gated channels. The big one that we want to focus on, we really want to focus on this sodium channel. These voltage-gated sodium channels. And make sure I would honestly write it like this: voltage-gated sodium channels open due to the spread of cations from the ligand, I would even call it from the acetylcholine ligand-gated cation channels at the motor end plate. Okay, so when these voltage gated ion channels open, we get a bunch of sodium rushing in, but some potassium coming out, but a bunch of sodium rushing in. And that is going to further cause the depolarization of the muscle cell, and it's actually going to spread it along, just like we saw in the, I believe it was this lecture, when we were looking at neural cells. Let's find just like here, how it spreads and it goes across like this and continuously moves. Same thing is happening inside of the muscle tissue. And that is actually the excitation in the muscle tissue. We're going to stop this video right here, and we'll see kind of what happens after this point.